Hey everyone, welcome to today's review session. Today we're going to be covering gene expression regulation. But before we go on to gene expression regulation, I know that some people had trouble or have been struggling a little bit with some of the concepts from this earlier week. So just a quick review of signal sequences. This graph or this chart that I found from last semester was really helpful, I found. It shows a really nice kind of flow chart of how, I'm gonna put the pointer on, of how signal sequencing works in the cell. So of course you start off with the mRNA and then the mRNA binds to a ribosome. That ribosome causes translation to start since ribosome is what causes translation. And so you go into the ER signaling sequence, and if it has an ER signaling sequence in the translation, then it will go on, of course, to dock at the ER, in which that polypeptide protein will enter during translation. And then you continue on. If it has a signal anchoring sequence or that transmembrane domain, it will remain embedded within the ER membrane. From there, it could be transported to the Golgi via vesicles if need be. If there are other signals, it can be either retained in the Golgi, go back to the ER, go out to lysosomes, etc. If it does not, or if it didn't have that anchor sequence in that first place, if it instead had a quote unquote move on sequence, it could go onto the ER lumen, into the Golgi, and then continue onto the plasma membrane, where it can then either be embedded within the plasma membrane or be secreted out of the cell to do whatever it needs to do, whatever its job is, go to its target. If we go back up, and if it didn't have an ER sequence in the first place, it would be continued to be translated by a free ribosome instead of one of the ribosomes at the ER. And then if it has another sequence, it could go to the nucleus, again, fully folded. It can go to the mitochondria or chloroplast. We don't really talk much about the chloroplast, but essentially any sequence to localize those RNAs. If it does not have those other sequences, it would just remain in the cytoplasm. So these are some practice questions. This is a recording, so obviously no one is going to be answering me, but we can just go over them as a review. So a protein has an ER localization sequence and an NLS. Where will it be located shortly after translation? If you guys want to pause and think about it, by all means go for it. But I'm just going to say the answer now. So the protein will actually end up in the ER because since it never gets the chance to bind to important, it has this NLS, but it also has the ER localization sequence, which comes before the NLS. So it will still end up in the ER and not bind to the important to even get into the nucleus. Number 2, a protein has an NLS and an ER retention sequence. Where will it end up? In this case, it would end up in the nucleus because even though it says that it has an NR retention sequence, it doesn't say that it even had a localization sequence. So a retention sequence is useless if it could not get to the ER in the first place. So instead, it would bind to the important to be imported into the nucleus. Number 3, a protein has an ER localization sequence and a mitochondrial localization sequence and a Golgi retention sequence. Where will it end up in this case? In this case, it will end up being retained in the Golgi apparatus or the Golgi complex. So essentially, the ER localization sequence comes before the mitochondrial localization sequence. And so it will be brought to the ER immediately since it goes there while it's still being translated. And then once it gets into the ER, since it has the Golgi retention sequence, it will then move on to the Golgi where it will be retained and staying inside the Golgi. Last question, a protein has a NLS and an NES. What would happen? So in this case, oops, don't know what happened there. In this case, the protein can move in and out of the nucleus. So because it has the NLS, it can be imported into the nucleus. And then because it has an NES, it can be exported out of the nucleus. All proteins that are able to go in and out of the nucleus must have both the NLS and the NES. If it has one or the other, it can either only go in or it can only come out. It wouldn't be able to do both. So since this has both, it can move in and out of the nucleus need be. All right, and then nuclear import-export as a recap as well. I know a lot of people were a little confused about this. So what I found really helpful is to use the names of the GEFs and the GAPs. So GEF stands for guanine nucleotide exchange factor, and you could think of it that it exchanges the GDP for GTP. And how it does is that the GEF will bind to RAN. In our case, it's RAN, but it can be any GTPase in this case. Which causes a conformational change in RAN. And then this conformational change or change in shape is going to reduce RAN's affinity for GDP and disassociate. So GDP is now out of RAN. And then since there is a lot of GTP around the cell, it can freely diffuse into the binding pocket of RAN. So now the final outcome is RAN bound to GTP. And this is all going to happen inside the nuclease. Now the GTP RAN complex is active. And then if you look at GAPs, it's the GTPase activating protein. And activating, you want to, in this case, associate it with hydrolysis, in which GAP can hydrolyze GTP to GDP. So from a triphosphate, you hydrolyze it and one phosphate goes away, and now you get GDP, the diphosphate. And this does it because RAN itself, since it is a GTPase, it has that activity where it can hydrolyze GTP on its own, but it's very, very slow. So definitely slower than a cell needs it to function. So with the help of GAP, it can increase the rate of reaction and hydrolysis is going to occur faster so that RAN GDP can be inactive again to bind important and such like that inside the nucleus. But this activating protein occurs in the cytoplasm. So nuclear import specifically, we can look at this chart over here. It starts off with your important, which is unbound, and then your cargo protein, fully folded. Since it goes through the nuclear pore complex, it's big enough for it to be fully folded. But the protein that has the NLS sequence right here, and that's just showing this, will bind to the important. And then it's going to travel through the pore complex, the nuclear pore complex, and go inside the nucleus. Over here, you can see that GEF is doing its job by exchanging the GDP for GTP. And then this causes the conformational shift so that the cargo is released and that the important can bind into the RAN GTP complex. And as a whole, this entire complex will now come back out of the nuclear pore complex, through the nuclear pore complex, back into the cytosol, where GAP is going to play its part and activate or help hydrolyze faster the GTP to GDP. And you can see this hydrolysis happening where that extra phosphate is going to leave and probably be recycled, do whatever it needs to do inside the cell. And then the entire cycle continues. And then the opposite or the reverse is nuclear export. So we're going to start inside the nucleus, where you have a cargo protein with this NES sequence that wants to get out of the nucleus into the cell or into the cytoplasm. So you have exportin, and all of this needs to bind together to leave the cell. And to do that, first, RAN GTP, again, it becomes GTP from this guanine nucleotide exchange factor. So it becomes GTP that allows exportin to bind, which then allows all these conformational shifts for the cargo protein to bind. Together, they come out of the nucleus into the cytosol, where GAP does its thing. It hydrolyzes from triphosphate to diphosphate. And now you have that extra phosphate here. But due to this hydrolysis, conformational change occurs again, in which the cargo is released to go do its job, find its target, whatever it needs to do. And then that exportin is also released to go back into the nucleus so that it can export out more proteins, be recycled, do what it needs to do. All right, and then just some more practice questions that you might see or might be really helpful for problem sets or future exams. So true or false? GEFs are located in the nucleus during nuclear import and export. This is true. GEFs are located in the nucleus during import and export. They are what helps GDP become GTP again, that exchanging factor. Number 2, GEFs phosphorylate a GDP to make GTP. This is false. GEFs do not phosphorylate anything. They don't add a phosphate to make GDP become a triphosphate. It just simply exchanges it by disassociating the GDP, and then GTP will go into the binding pocket. Number 3, GAPs stimulate a RAN to hydrolyze GTP to GDP in the cytosol. This is true because it will that RAN's intrinsic activity to hydrolyze. GAP just helps it move faster. But of course, you want to always be careful of trick questions. If it said in the nucleus instead of in the cytosol, then in this case, the answer would be false. Number 4, GDP is in higher concentration than GTP. This is false for sure. In the nucleus, the whole reason why if you just disassociate GTP will go in is because there is way more GTP in the nucleus than GDP, so much more so that if there is a RAN that does not have GDP bound, GTP can just bind in easily by itself. And then we can go through these as well really quick. What would happen if the KD between important and the cargo protein was greatly decreased? And so if you think about it, the important cargo protein complex would be stuck together and likely cannot leave the nucleus together, because this is similar to a loss of function mutation in the GEF. Number 6, what would happen if the KD between exportin and the cargo protein was greatly increased? And then the answer to this would be that the exportin and the cargo protein complex would not be able to stay bound. The cargo protein would not be able to leave the nucleus either. The exportin instead can still go through its cycle, but it just wouldn't move the cargo protein with it. Remember, KD is the dissociation constant. So if you have a really high dissociation constant, that means exportin and cargo likely would not be able to stay bound together. Number 7, what would be the result of a loss of function mutation in RAN-GEF? This is really similar to all of your problem set questions. So essentially, RAN would no longer be able to replace GDP for GTP because the exchange factor is not working anymore. So in the import cycle, this would prevent the release of cargo proteins from importin. And then in the export cycle, the exportin cargo complex would not be able to come together to leave the cell. Or not leave the cell, leave the nucleus, sorry. All right, so we are going to move on to gene expression and regulation, the newer part or the last part of this week's content. So gene expression is the production of proteins. Pretty straightforward, but it's basically very carefully regulated in cells at all levels. So throughout the entire central dogma process, gene expression is very carefully regulated inside the cell. And so it's a brief review of the central dogma. Eukaryotes, you have transcription inside the nucleus and then translation outside the nucleus in the cytosol. However, in prokaryotes, transcription and translation are done at the same time in the cytosol. We'll definitely go into more detail about this later on in the course, but this is the general knowledge that you need to know right now if you've forgotten from intro bio or any of that. And so to continue the central dogma theory a little more in depth, so at first DNA is read by the RNA polymerase to make mRNA. This is known as transcription. So you have your DNA, RNA polymerase comes in to then make mRNA. And then the ribosome is read by the ribosome reads the mRNA to know in what sequence the amino acids would be strung together. And then this is translation. So you have your mRNA, ribosome comes in, reads the mRNA, gives it the amino acid sequence. And then from there, like we learned last week, due to R group interactions, the protein folds into specific conformations in structure equals function. So whatever its conformation is will help the polypeptide do its job. So just a quick another practice problem to help you guys get a better understanding which one of these are involved in gene expression regulation. So if you wanted to pause, go ahead, think about it. But the answer is all of them. There is definitely a lot of ways that gene expression regulation is done inside the cell, and these are all various ways that the cell does so. And then we'll go into a little more detail and explanation about each one. So eukaryotes and prokaryotes are certainly different, but if you understand the gene expression regulation in eukaryotes, you will be able to understand the mechanisms in prokaryotes and along with some of the differences. So we'll start with transcriptional regulation. This is the main site of regulation of gene expression. It is definitely a huge way that it's definitely a huge way that cells maintain or regulate gene expression. And it starts off with transcription factors that regulate the RNA polymerase binding to promoter DNA. Without transcription factors, RNA polymerase cannot bind to DNA and starts transcription. So if you look at this picture, you have all these different transcription factors, and then RNA polymerase binds to the transcription factors to then bind to the DNA promoter. If these transcription factors weren't here or something happened to them or there was something blocking the way, then this RNA polymerase would not be able to come into the DNA to bind, and then transcription would be very downregulated, or it might not even happen at all. And these are also more specifically regulatory transcription factors. They are very gene specific. So certain transcription factors will go to certain promoters or certain areas of the DNA. And so you can have positive or negative regulators. A positive regulator will enhance transcription when bound to the DNA. You can think of it as the more RNA polymerase is able to bind, then the more transcription you have happening, and then the more that the gene is expressed. On the flip side, you have negative regulators where it will essentially suppress transcription. So the opposite, the less that RNA polymerase can bind, the less transcription that will happen and the less gene expression you will see. There's another way that transcription regulation can occur. It's through chromatin. Remember that prokaryotes do not have chromatin. They don't have histones, but eukaryotes do. So chromatins are when the histones are wrapped in DNA. So these orange circles represent histones, and then the green lines are your DNA. And so this packing can determine which genes can be expressed. So chromatin has two forms, as you can see in this picture. You have euchromatin, which is a very open state. so to say, in which the RNA polymerase and transcription factors can bind to DNA. Since it's very open, there's a lot of space for RNA polymerase, all the transcription factors to get in to transcribe. However, the heterochromatin is a much more closed state. They're very packed tightly together. And so because of that, RNA polymerase and the transcription factors cannot reach the DNA or bind in, since they're too tightly packed. And the outcome would just be that genes cannot be transcribed, since the RNA polymerase can't bind in the first place. So RNA modification and processing. RNA has to be processed and made stable into mature mRNA in order to leave the nucleus. So when you first get mRNA, that's your pre-mRNA, and then it has to be processed and become more stable in order to mature and leave the nucleus. And this is because when you leave the nucleus, there are a lot of aspects inside the eukaryotic cell that can endanger the degradation of the mRNA. And so how does the RNA become more stable? And it's through the 5' cap and the poly A tail. The 5' cap over here, as you can see, it's called that because it goes onto the 5' end of the DNA, but it is covalently added to the 5' end. And so, oh, sorry, I said DNA. It's to the 5' end of the RNA. And this essentially prevents the RNA molecule from being chewed up by the nucleases. Nucleases are enzymes that can degrade or quote unquote chew away at the molecules, at the RNA molecules. And this is done so because the 5' cap, these are guanosines, but because it's a special type of G, essentially, it's almost like it's flipped. And so because of the different structure, it physically cannot fit into the nuclease active site. And if nuclease can't bind to this end, it can't chew away at it. The poly A tail protects the mRNA from being degraded. So it's just a lot of A's at the end, at the 3' end. That's why it's called the poly A tail. And it's not that this tail itself helps protect the mRNA from being degraded. It's actually a bunch of different proteins that can bind to the poly A tail, and those are what protects it from being degraded. Any and all mRNAs, though, will be degraded over time. So the poly A tail just kind of prolongs the process, gives it a longer life. But they will eventually be degraded because the poly A tail will slowly be chewed away at. All right, so another way of gene expression is RNA modification and processing. Oh, sorry, we're already on this, but it's RNA splicing. So you have the 5' cap, and then you have the poly A tail. But another way that RNA is continued to be processed is through splicing. So all RNAs in eukaryotes have these non-coding regions. They're called introns, and they have to be spliced out before translation in order to leave the nucleus. So introns, again, they're just areas that are transcribed, but they are not translated since they're cut out before translation, before they leave the nucleus. Then on the opposite end, exons are the areas that are transcribed and translated. So in this picture, the dark parts are the exons, and then the uncolored spaces are the introns. You can see that the introns are spliced out, they're taken away, just left out, and then the exon colored parts are put back together, and now you have your mature mRNA. And the enzyme that does this process is called a spliceosome. There's also another mechanism called alternative splicing that Professor Francis had mentioned. It's also known as exon skipping, and it basically allows the cell to regulate which exons can make it into the final mRNA transcript. And it's really useful to get more diversity out of one protein, out of one single gene. So a single gene can make different types of proteins. To give you a little better understanding of what alternative splicing is, so say you have these different exons, and then you take out all the introns, which were these green parts. So one way to put the exons together might be to take 1, 2, and 3. So you splice out number 4 as well, or you can splice out number 3, and then you can get a protein that's 1, 2, and 4. And then this is a little, another picture. So you can have all these different exons. And then if you splice, you can get this combination, or you might get this combination, and then structure equals function, of course. So these different types of combinations will then produce different types of proteins. And then some other ways for gene expression is that the cell can also stabilize mRNA, or basically control the mRNA degradation rate. So yeah, and mRNA's lifespan can be regulated. This involves RNAi, or RNA interference. We haven't talked about this yet, but it'll be something we'll talk about later in the course. And so something important to know is that cells can control the overall mRNA degradation rate. So it can control when the mRNA are going to be degraded throughout the cell, but say that something in the environment changes, and so they really need a specific mRNA to be degraded now or immediately, the cell can certainly also target those specific mRNAs to be degraded when it needs to. Number 4, translational control. This is essentially, like the word says, controlling translation, controlling how often or when or how easily translation can occur. So from the RNA to protein, how easily that process can occur, if at all. And you can control this by a couple of different ways. One is our ribosome binding accessibility. So you can either block or help the ribosome. A protein can do that. And if you block the ribosome from binding, this will definitely decrease the ribosome binding. If you decrease ribosome binding, that means that translation cannot occur, or it will occur a lot less. If you help ribosome binding, it'll definitely increase how often ribosome binds, and then that in turn enhances translation. Another way that translation control can occur is through mRNA localization. And this is essentially where the mRNA is located can affect gene expression rate. So is it going to the ribosome? If the mRNA is going to be localized to the ribosome, of course, that will allow for translation to occur. If it is maybe going to be localized away from the ribosome, sequestered away from the ribosome, that means the ribosome can't get to that mRNA strand, and of course, no translation will occur. This also involves RNAi, so it's something that we'll talk about more in detail later on in the course. So we talked about the different types of functions or the mechanisms of gene expression regulation in eukaryotes, but we haven't really spoke about the prokaryotes, and that's again because the mechanisms are very similar for a lot, but there are just some things that because of the structure of prokaryotes and what they have, what they don't have, there are also differences, things that eukaryotes do that prokaryotes do not do. So to review a little bit, the differences here are that in eukaryotes, we have our DNA packaged into chromatin. So that's one way that we can regulate our genes. Again, remember that we can either, based on how tightly packed or how loosely packed the chromatin are, you can control how likely the RNA polymerase is able to get in there to start transcribing. But prokaryotes don't do this because they don't have histones. So there's no packing the DNA into chromatin or loosely packing the DNA, none of that. And then another difference is that since eukaryotes have a nucleus, the mRNA has to be processed and become very stable in order to leave the nucleus, so that nucleases and all that don't degrade the mRNA before it can be translated. But since there's no nuclease in prokaryotes, there is no eukaryotic-like RNA processing. They don't have to do that splicing. They don't have to make it super stable to leave the nucleus, since there is no nucleus in the first place. Prokaryotes do have some type of RNA processing. They do have a process to do so, but you definitely don't need to know that. It's beyond the scope of this course. If you're interested, maybe you might want to look it up yourself, but you don't need to know it for sure. And then another difference is that the eukaryotes can regulate transcription with transcription factors, and prokaryotes can do so as well. The only difference is that prokaryotes and eukaryotes have different types of transcription factors. Eukaryotes have a basal transcription factor, whereas prokaryotes use a sigma factor. A sigma factor is a type of general transcription factor, but in bacteria. And so, sorry, but yes, they both have transcription factors. They both need the transcription factors in order for RNA polymerase to come in and bind to start transcription. But it's just that the types of transcription factors they use are different. So then there are similarities just as much as there are differences, of course. So they both regulate mRNA degradation rates. They both regulate the mRNA localization, so where the mRNA goes or where they are, whether it's to the ribosome or not. They regulate the rate of translation as well, along with the protein activity and degradation. And then this is just a visual. I think this was seen in Professor Francis's video lectures. So for eukaryotes, you have the starting with the DNA, transcriptional control. Now you have the RNA transcript. You process the RNA, and now it's ready to leave the nucleus. The mRNA transport and localization control, will it go and be degraded maybe, or is it going to go to a ribosome to then become a protein? That's where translation control occurs as well. After proteins, you now have post-translational control because it's already translated into a protein. Post-translational control, I didn't have a slide on it, but we covered that last week in week 2. That's where you have all those covalent additions, so acetylation, phosphorylation, methylation, all that, ubiquitination. That's one of the ways that proteins can be degraded. There's also things like proteases, which are enzymes that can break down proteins and polypeptides. So the cell can definitely regulate all of those aspects that I just mentioned to either maybe degrade proteins or make them more active or make them inactive, so on and so forth. And then the difference between that with prokaryotes are that transcriptional regulation prokaryotes do have, but it is certainly very different and not exactly on the chromatin control level. They do something, it's just not involving the chromatin since they don't have histones like eukaryotes do. RNA processing control, they process their RNA, just very, very different for prokaryotes. You don't need to know how, but they do process it, just not the same way that eukaryotes do. And then there's no need for prokaryotes to stabilize, transport, localize, all that. Well, they localize, sorry, but they don't need to have that stabilizing mechanism because there is no nucleus here. It's just all cytoplasm. And then everything back here is more or less the same mechanistically. And yeah, so that reaches the end of this review session. Hopefully I didn't go too fast. Feel free to slow me down or speed me up, I guess, 2 times speed, 0.5 speed, whatever you need to do. I didn't want to make this too long of a video, but hopefully it was a good pace and that you guys still understood it. But thank you guys for watching, and definitely feel free to reach out if you have any questions. We're always here by email in any way that you need. And then these are just some quotes that I found that were corny and sciencey, but ignore those if you want. They're really corny. But yeah, thank you for watching, and have a great weekend, guys.